How are you? Do you hear me and have slides or no? Please say to me and I want continue. Do you have do you hear me and have my slides or no? Yes. Last class done was not work. Okay. We want today speak about introduction of microbiology and what is microbiology means. Microbiology is the biology of microscopic organisms. Microbiology should be an easy word to define. Two, science or logy of a small or microorganism. They study in this field you study living of this small that they cannot be seen with the naked eye. It means if you want a study of this, you should use what we should use. Do you know what we should use? Do you know? We should use microscope. Field writing, okay? Okay. Microbiology is the study of microorganisms usually less than 1 millimeter in diameter, which requires some form form of magnification to be seen clearly. Yes, in one microscope. Such virus, bacteria, and archaebacteria, fungi, protozoa, yeast, and microscopic algae. Please write this name of microorganism. This is kind of microorganism. Please write. Let's I don't know what should I do, Haydar. I don't know. I want teaching you. I don't know what should I do. Really, I don't know. Okay. The importance of microbiology in the world today, just consider the following list of some of the general areas which the experts of microbiologists might be used. What? Environment science, food and drink, fundamental research, agriculture, pharmaceutical industry, and genetic energy. Please write this. The space or field the microorganism or microbiologists can study microbes. Okay, do you hear me? Okay. Benefits. What is benefits of a study of microorganisms? First, genetic engineering. Learning about synthesis of chemical products. Recycling storage. Bioremediation or use microbes to remove toxins, means oil spills. Use of microbes to control crop pests and normal microbacteria. Please write this. The field of benefits of microorganisms. The sun intermittent and another. reach us completely. Okay. Harmful effects. What is harmful effects of microorganisms? Cause disease means basis of bioterrorism. Food spoliation. It means cause disease basis for bioterrorism or food spoliation is base of or case of microorganisms. Pioneers of microorganisms. Okay. Do you hear me? We should learning about. Okay. Robert Hooke. Robert Hooke. In year 1065 proposed the cell theory. How? He observed cork with crude microscope. All living things are composed of cells. Okay. What should I do? What should I do? Do you hear me? Fatima, do you hear me? Robert Hooke. Okay. He see microorganisms living cell in his microscope. How? Come here. With this, I think what do you mean, Ali Hamid? I should learn you and I can't. I don't know. Come on here. I should learn you. I can't interrupt. I should continue. I don't know what you mean. Okay. Louis Pasteur. He lived in 7022 and 108,095. He was chemist. He studied or he explained about fermentation and then pasteurization. What do you mean? He learned about heat liquid enough to kill spillage bacteria. And then he say about vaccine development on rabbits and proposed the germ theory of disease. The germ theories of disease. He proposed aseptic techniques prevent contamination by unwanted microbes. Director of Pasteur Institute. He was director of Pasteur Institute. He was one of the famous and important person in microbiology science. Okay, do you know? Continue. Do you want right or I continue? Koch, Master Koch. He was the specific causative agent must be found in every cause and of disease. He say one specific causative agent must be found in every cause of the disease. The disease organism must be isolated from the lesion of the infected cause and managed in pure culture. The pure culture inoculated into a susceptible or experimental animal should produce the symptoms of disease. The same bacteria should be isolated in pure culture from the intentionally infected animals. Please write, this is very important, Koch postulated. Please write this slide. It is important. This is examination. This is one field of your exam. Thank you. When you write, I will continue. Okay, you right? Continue. Explain what is the nature of question in the exam. Okay, I will tell you. Okay, I will tell you. I give you a explaining exam. You should write about what the meaning of microorganisms, such this. Okay, I will explain you. And my exam is from this slide, only this slide or file I will give you. Okay. Okay, medical microbiology. Medical microbiology is the study of microbes that cause disease in humans. Microbes are everywhere, both within the outside or body. Most are harmless. We will be ill for us or will be disease for us. Such as, do you know what medical microbiology? Can you explain? What microbiology, medical microbiology? Coxy. Which kind of fox? Streptococcus, you mean? Okay. Like mycobacterium. It means cell. I don't know what they say. Okay. Type of microbes. What one kind of our microbes are eukaryote, and both of them you know, all of them are prokaryote. Eukaryote organs. These include fungi, means models or yeast, parasites, means helminths or protozoa. They have a complex cellular structure similar to those of human and animals. Their cells have nuclei and mitochondria, and they are largely self-sufficient and capable of independent life. Okay, right? And then tell me, I will continue. Okay. Fungi. What is fungi? This is, they have a sporangia. They are eukaryote, have kitten cell walls, please write. Use organic chemicals of energy. Molds and mushrooms are multicellular, consisting of mass of mycelia, which are composed of filaments called hyphae, and yes, are unicellular. Please write the, okay, and it is very important. It is very important. The masses of mycelia and they have hypha. This is hypha. Hypha. Yes, are unicellular. Okay, please see. When you write, please say me. I think only Fatima Adnan is in class. Another person they aren't, like Diana Roya, Ali Hussein Naji, Allah Tafi, Afia. Where are you? What you don't say anything? Uh-huh. You come, you found. Okay, right. I want continue. Okay, let's continue. Protozoa. they are eukaryotes. They absorb or ingest organic chemicals. Maybe maybe motile via pseudopoda, cilia, and pelagia pseudopoda must freeze some parasites. Feels right? Okay. Algae. They are eukaryote. Cellulose cell wall. They have cellulose cell walls. They use photosynthesis for energy. It means primary products. Okay, continue. Feels right. Okay, that I told you was were eukaryote bacteria which are eukaryote. But I want to explain you about prokaryote organisms. These are simple and large cell recipients, unicellular organisms which have no nucleic and internal diving membranes. What are usually happening walls contain mucopeptides which tell us peptidoglycans. They are collectively named bacteria. Some genera, Rickettsia and Chlamydia, are not capable of dependent life and are therefore named atypical bacteria. The latter group of organisms is obligate intracellular pathogens, which require and presence of viable eukaryote host cell for growth and reproduction. Please write this slide. These are very important for your exam. Write and then tell me continue. Yes, I wait. Fatima, I wait. Fatima, continue or you want to write? I think only Fatima write. Yeah? Continue. Okay. Bacteria. Classic bacteria, these organisms produce asexually by binary transverse fission. Do you know what kind of fission or what kind of, methods? They have methods. They are not pulses to nucleus typical of eukaryote. You remember the chromosome is inside of cytosol. The cell wall of these organisms are rigid with some extensions and microplasma. Oh, not important. Feels right? Like what? Like this bacteria, like bacilli, like staphylococcus aureus, like mycoplasma, like pseudomonas, like enterobacterias. Okay, feels right? What? Ah, yes. It's like Neisseria, gonorrhea. Feels right? I will continue. Okay, continue. Chlamydia or rickettsia. These organisms are obligate intercellular parasite road-shaped to cocci. Mycoplasma are bacteria without rigid cell walls. They have one layer, and very big and hard layer of lipid, mycoplasma. They have hard layer of lipid on their cells. What do you see? Uh-huh. It isn't important, Haidar. Uh, I know you're not. It isn't important, Haidar. I know. You can't do anything. and I should teaching because we having class last week. We should have class this week. Okay. Virus. A cellular. They haven't. Please attention. They haven't cellular organisms. They have only DNA or RNA, not both of them. Only DNA or not or DNA, RNA. Core is surrounded by a protein coat. Coat may be enclosed in a lipid envelopment, and viruses are replicated only when they are in. Okay. When you write, tell me, I will continue. You don't have any question? Why you don't have any question? It means core means DNA or RNA. The base of virus don't have membrane or cell wall. It have only one protein coat. You understand, Fatima? Okay. Next slide. Okay. Core means Okay, multicellular animal parasites. They are eukaryotes. Their parasite platforms and roundworms are called helminths. They have microscopic stage of their life cycles. Please write. Okay. What do you mean? No, it is not disturbed. Okay. Let's go next. Please look here. This is one eukaryote cell. Here, right? You see eukaryote. They have nucleus and mitochondria and endoplasmic lysosome. They have mitochondria, Golgi, this vesicle, plasma membrane. You see here, most of organs they have. Most of organs they have. In eukaryote, you have most of organs. Sorry, we have one cat in our home. We called him Mahman, and he come to my room because he was very hungry. Sorry, because he's not his voice and disturb you. Okay, next slide. This is prokaryote cell. Prokaryote cell. It hasn't nuclei. We have a nuclei here. We have chromatin or chromosome this side here, and they have ribosome only. They didn't have mitochondria or ribosome or Golgi or ER. They have cell wall, but in eukaryote you haven't cell wall. Do you hear me? Someone please answering me. Fatima. Uh-huh. And we are here have membrane, two bilayer membrane. We have phalangia, cytoplasm, plasma membrane. Okay, next slide. You don't have any question? This is one prokaryote cell structure. This is one bacteria. Means maybe bacil because of this shape. This is the chromosome formatting, you know. They are pili. They are pili. Here, pili. This is pelagia. Pelagia is useful for what? For what? What work bacteria will do with pelagia? You know? Yes, for moving. Okay, for moving, yes, that's right. Here is what kind of cell? Please tell me. This is eukaryote or prokaryote? Do you know what kind of cell are these? You don't know? They are prokaryote cells. Roya, what do you want to say? Okay, okay. Right now it finished my slide. I should load for you another slide. We have some rest, maybe 10 minutes. And our class is until uh, 6:30, yes? And now, what time we should finish? Okay, okay. Please, you can have some rest. I want uh, change my slide. Okay, some 5 minutes we have rest. have this structure. Flagella, pili, capsule, plasma membrane, cytoplasm, cell wall, lipopolysaccharide, tychoic acid, inclusion and spores. Please write. Okay, next slide. Okay, for classification of bacteria, one method is base of reaction to dye. Please write. They have two categories. Maybe they can gram positive. What color you see in gram positive bacteria? You know what color? I told you in first section. What color? Gram positive what color? Yes, perfect. And in gram negative, what color is in negative? Gram negative? Okay, Roya, thank you. Gram red or pink? Yes? Yes. Pink. This method of staining named after Kirsten Graham in 1884. Please write. Please write. Okay, next slide. This is gram X-ray. At the first, we draw crystal violet on our microorganisms. And then here we use logo. Then alcohol wash, and end we use saffron. 4 groups based on cell wall size of the cell. Okay, it is okay. Fatima, it is okay. Yes. Based off the cell wall composition, we have 4 groups of bacteria. First, gram-positive cells. Please write. Then, gram-negative cells. Bacteria without cell walls, like Mycoplasma. Bacteria with chemically unique cell walls. Okay. Next. Do you have any question? Okay. Bacteria with chemically unique cells. What kind? They don't have gram cell walls. It means they don't have peptidoglycan cell walls. They have lipid cell walls. They have lipid cell walls. Or their cell is acid fast, like Mycobacterium grams. They are gram-positive type of cell wall, but lipid. It means mycoloic acid, waxy substance, does not decolorize. Okay, please write. I want to continue. Next slide. Okay. Shape of bacteria. This slide explains you shape of bacteria. We have bacteria in three shape, three main course, main shape. One is cocci, like this. One is cocobacil. One is bacil vibrio, which is curved, and Aspergillum spiracat. Okay, please look here. Cocci are in two types. They may be chain. It means a streptococcus, or cluster like staphylococcus. Please write. When right, please tell me, I will continue. Okay. Please see here, they are cocci. Okay. Cocci, cluster, diplococcus. Streptococcus, we told you they are cluster and they are gram-positive. See here in continue in microscope by gram. Okay. This is a staphylococcus, like this. A streptococcus. Okay, please wait. I told you about this. Okay. RNA sequence. The target, the greatest advance in classifying organisms is RNA ribosome or ribosome RNA. Different nucleotide sequences are used to classify prokaryotes. Prokaryotes have two rRNA. One is 60S, it's called 60S. One with say 23S. Please write. Here, please see. Okay, this is our RNA or ribosome RNA. One part of this is CS. This is in prokaryote. Please see. And one is heavy subunit we call panjo-S. But in prokaryote. But in eukaryote, we have 40S and 16S sub. Okay, didn't see? Okay. In a strain, what different with gram positive and gram negative? Gram positive have big layer of replicator and take crystalline dye on this cell. But gram negative, they haven't replicated and they can't take crystalline. Do you understand? Do you understand? Do you have any question? A slide is important. You should write. In peptidoglycan, we have 4 amino acids. Texture possible flag operation in yes, again. Here, no, here. Here, do you see here? Rata Fatima? Do you mean this? Okay. Okay. What time we have class until when? Until 7 or half past 6. You know? Okay. Gram negative, I write for you. No. Our class is until 7 or half past 6. Please say me. Okay. Do you understand? Okay. Let's go. Okay. This is very important. You should write this. This is peptide layer. Okay. Peptide makes from four amino acid. They are in a strain, in chained together with L-alanine, D-glutamine, L-lysine, D-alanine. There are four amino acids. One side they have polysaccharides such an acetylmuramic acid, and another side they have an acetylmuramic acid. Please write this. This is very, very, very important. Please write. I'm waiting for you. Please write. This is very important. My friends, please write. Yes, yes, you should save and you should now. Please write. Please write and tell me continue. Okay. What do you mean, Marvel? No, no, save no. Please write, please pray. Please write. It is important. Okay. Okay. Next. Here you see peptidoglycan. Peptidoglycan have L-alanine, D-glutamic acid, L-lysine, D-alanine. In gram positive, these are amino acid. But in gram negative, beside L-lysine, we have D-amino palmitic acid. You know? Okay? Okay. Please see here. This is peptidoglycan in gram positive. Or this is peptidoglycan in gram negative. You can see the line. You know? Okay, next. Here, gram-positive cell wall, peptidoglycan, gram-negative cell wall, peptidoglycan. Okay, this here, in gram-positive, we have tychoic acid. Tychoic acid. Tychoic acid is from glycerol, phosphate, and ribitol. And this is attachment from fish. Please write. Okay. Here, there are tychoic acid. Up of peptidoglycan. Peptidoglycan. This is a peptidoglycan. Tychoic acid is up of this. Upside of this. Next. What's the function of tychoic acid? They are antigenic determinant. They are predictable of supply of Mg to cell by binding Mg2 positive. You can't. Why? Wait. Is it okay, Fatima? Is it okay, Fatima? Fatima, is it okay? Okay. They are antigenetic determinant. They are partial of supply of Mg to the cell of Mg. تماس میگیرم سر کلاسم. There are regulated normal cell division. For most part, proteins is not found as coincidence of gram-positive cell wall expect. M protein on group streptococcus. Please write this. Please write, and then I continue. Okay. Uh, are you tired? Are you tired? We will continue in the next lecture. No, yes, 50 minutes, only 50 minutes, until 7. Okay, okay. Finish, because you are fasting. I know. Because you are fasting. We finished. Yes, ma'am. Because you are fasting, we finish. Please pray for me. Yes, I know you are fasting. Please pray for me, my dear friends. I really need. Thank you, Haida. Thank you, Holly. Thank you, Paul. Marvel.